of the year from our microbiology group, from the Multi-ômicas group here from Unica. And it is my great pleasure to introduce Dr. Tan. She is assistant professor from City University of Hong Kong. She holds a PhD in civil and environmental engineering and bachelor's degree in biological sciences from Nanyang Biological University from Singapore. Amy works at the interface of biology, engineering, and information science to harness the knowledge and power of microbes for waste to energy and bioresources applications. So thank you so much, Amy, for accepting our invitation, and especially for the effort, because now it's 8 p.m. in Hong Kong. And so thank you so much again, and the floor is now yours.
Oh, thank you so much, Henrique, for inviting me, and it is my great honor and pleasure to be sharing with you the work that we are doing here in Hong Kong, and something about a topic that I'm very passionate about, and hopefully we it can be inspiring to everyone here, and then we can have a nice discussion afterwards. So maybe let me share my screen first. Can you see my screen?
Yes.
Okay, great. The topic that I will be talking about today is big.
It is not showing your presentation, Amy. It's showing
It's not showing. It's showing something else. Okay, let me see how I should adjust this. Try again. Maybe touch screen. Does it work now?
Yes.
Yeah. You can see the PPT. Okay, great. Okay, so the topic of my presentation today is a big data approach to study tiny microbes and break classical limitations in waste valorization. So I'm sure all around the world, we are very concerned about how we move towards sustainability and also transiting to carbon neutrality. And this is also true for Hong Kong. So just to give everyone a little bit of background on what we are doing here in Hong Kong. Hong Kong actually has this Climate Action Plan 2050, which really outlines how we are going to be transforming the energy and resources landscape in Hong Kong. And in this Climate Action Plan, it talks about tangible goals to achieve zero carbon emission by 2050, to develop a livable and sustainable city, and to reduce fossil fuel reliance and carbon emission by 35% by 2030, and increasing this reduction heading towards 2050. So overall, the agenda of moving towards a renewable energy, renewable bioenergy, as well as biomaterials and bioresources is a very important agenda for Hong Kong, and I believe also in Brazil as well. So a lot of what I do here at CityU pertains to that. Our research is really focused on using organic waste and wastewater, and then turning them into valuable resources like bioenergy, bioresources, as well as clean water. And the way we do that is through bioprocesses. And at the core of bioprocesses, of course, microbes are the key players over here. So our research focus, one of our research focus is using pan-omics approach to find Goldilocks conditions for surpassing classical limitations. Because a lot of the technologies that we see these days that are in use at sewage treatment works or organic waste facilities are already approaching their performance limitations. And the traditional approach of adjusting operation conditions, reactor design, could only improve the reactor performance by a little bit, or maybe not too much improvements at all. Therefore, we want to seek to find ways to understand the microbes, and then hopefully through understanding microbes and what makes them tick, we can try to enhance treatment efficiency and performance and increase resource harvesting from waste so that we can push the performance threshold of these technologies towards more bioenergy production, thinking about even on-demand bioresource production, and even generate new processes. So, what is pan-omics? And pan-omics is this idea where instead of just using a single layer of information like genomics or transcriptomics to understand biological system, pan-omics actually utilize multiple omics layer in conjunction in order to gain deep insights into the underlying biological mechanisms. So this is just a very nice picture of pan-omics illustration that I got from this textbook. And it really shows the way genetic information flows from DNA towards RNA, proteins, and then bringing about phenotypes that we see in any biosystems, be it a single living cell, a bacterial cell, be it us, or even biosystems. So what can pan-omics actually tell us? Its individual layer. For example, at the genomics layer, which is basically the blueprint, the genetic blueprint of any living organisms, we can use information here to tell us for a biosystem, what are the microbes that are there in this system? This same information can also be used to decipher what are the genetic pathways or metabolic pathways that are present within each of the microbes. And through transcriptomics, proteomics, and metabolics, we can then assess if the pathways that are there are actually active. So through this information, we can start to really construct a picture of how the microbes are actually interacting together. And this is especially true if they have trophic interaction in their metabolism, like in this case here, where bacteria A and bacteria B, they actually work together to effect this kind of biotransformation. And of course, all this will bring about the phenotype, which is what we typically see as a reactor performance and in terms of bioenergy production or bioresources production. In addition to that, with all this kind of knowledge, we can also further extend this information and apply it towards thermodynamics analysis, bioenergetics, flux analysis, network analysis, gene regulation, reactor modeling, etc., etc. So panomics is actually a very useful information and also a big body of information that we can tap on for various uses. So at our group over here, we are engaging on various research topics. And just to give you an idea on a few topics that we are working on, the first is we are looking at, or we have been working on actually increasing bioenergy harvesting from low-strength sewage. We also currently very active on the topic of increasing bioenergy harvesting from sewage sludge. And as an extension of my own PhD project, we are also looking into how to increase bioplastic production. So although these three topics are a little bit distinct from each other, but the underlying drive here is that we are trying to push the limitation of bioconversion, be it bioenergy harvesting or bioresource production. So in the interest of time, today I will only be focusing on topic 1 as well as topic 2. For topic 1, I will be sharing our panomics approach, which is utilized in a more traditional way. Whereas for topic 2, I will be sharing something a little bit more advanced that we are working on, where we apply quantum information theory to try to decipher gene regulation network. So I will actually spend a little bit more time on topic 2 and a little bit less time on topic 1, because I think topic 2 is a little bit more interesting. Okay, so for topic 1, this really actually started because our collaborators in Korea as well as in the States, they were looking at developing new technologies that could turn sewage treatment towards more energy neutral or energy positive. So I think we are all well aware that conventional sewage treatment is actually very energy intensive because it typically involves an aeration tank where air has to be pumped in for pollutants, COD removal. And this actually leads to very high CO2 as well as energy footprint. The energy consumption is about 40 to 60% of the entire sewage treatment process. And at the same time, this also leads to a lot of CO2 emission. So what our collaborators set out to do is they asked this question, is it possible to turn sewage treatment into an anaerobic process? And what they did was they came up with this very clever bioreactor design where it's actually a bioreactor with membrane inside. And then the bioreactor also contains biocarriers, specifically granulated activated carbon. So their idea is that this granulated activated carbon would serve two purposes. The first is it would act as a carrier to encourage biofilm formation and then to increase the overall SRT. And then the second is that this biocarrier can also act as a scrubbing effect to clean the membrane to reduce membrane fouling. So what they found is that they actually were able to get very good performance using just domestic sewage, which is low strength. So you can see over here, these are their published data where they were able to achieve 80% pollutants removal within 8-hour HRT. And because the whole system is anaerobic, so it also reduces CO2 emission by up to 70%. And most importantly, the nice thing about anaerobic process is that it always produces bioenergy. And based on their calculation, this biomethane that's being produced can actually turn the whole sewage treatment system into an energy positive system. They actually were able to get very good methane yield of 0.2 liter of methane per gram CO2 removed, which is about 50% of the theoretical yield. So this was very, very impressive. So one thing that was constantly at the back of their mind is what is it about their system that actually led to all this very, very good performance in terms of the COD removal as well as the methane formation. So that's when they started to get in touch with my then postdoc advisor at PolyU, Professor Henry Lee, and then we started to look into this together. So the hypothesis here is that perhaps the conductive material, the granulated activated carbon, could have something to do with this. So through experiments, they were able to verify this. So you can see that this is an experiment, a batch experiment, where they try to compare the effect of conductive material versus non-conductive material in terms of COD removal and methane formation. And solid squares and the circles here are actually duplicate experiments for the conductive material, and then the hollow ones are duplicate experiments for the non-conductive. So you could see that the conductive material is consistently increasing the kinetics of COD removal. And this is also true for the methane production as well. Methane production tends to happen earlier in the setup with the conductive material compared to the one without. So we knew that there must be something that's happening on the conductive biocarrier itself. So we started to look into the microbial community using just 16S rDNA community analysis approach. And something stood out quite apparent to us was that on the conductive material, which is this AFBrGAC and the AFCMBrGAC, we saw a disproportionately large population of methanocyta, which is traditionally known as an acetoclastic methanogen. It uses acetate for methane formation. And what is also interesting here is the coexistence of Geobacter in this community as well. And Geobacter is a very well-known exoelectrogen. It's able to actually metabolize substrates like ethanol and then produces electrons directly. So we think that the reason why this reactor is performing so well is that the conductive material is a surface where methanocyta and Geobacter is adhering to onto part of the biofilm community, and Geobacter is in fact transmitting these electrons directly to methanocyta that then uses these electrons for methane formation. And there is good basis to this because some other researchers have shown that the transmittance of electrons is actually 10 times faster compared to using chemical-based electron donors like hydrogen. So we really started to then focus on looking into methanocyta and to try to understand how is it able to take the electrons and contribute towards this enhanced methane formation. So using the metagenomics approach, we were very fortunate. We were able to actually obtain a good metagenome draft of methanocyta, and we were able to reconstruct the methane formation pathway. So this pathway over here is the acetoclastic pathway, where acetate comes in and then goes through this pathway towards methane formation. So this is the pink one. We also found that methanocyta has this photosynthetic-like pathway. Looks almost like the Kelvin cycle. And this pathway over here allows methanocyta to take in CO2 and then electrons to form formaldehyde, which can then be funneled into the methane formation pathway through methylene H4-MPT. So this is the second pathway, and then there is this other pathway here, which is similar to the hydrogenotrophic pathway, where methanocyta can use CO2 as well. But this pathway requires methanocyta to have an electron source. Since it cannot take hydrogen, therefore the electron source we believe must come from this electrons that's produced by Geobacter. So with this basis, now that we have constructed this pathway, we then use meta-transcriptomics data to check if this pathway was indeed active. And to our surprise, we actually found that all the three pathways here are actually actively transcribed, and they are all transcribed at about the same level. So this revelation actually leads us to the conclusion that the ability of methanocyta to take electrons directly is actually helping to overcome the classical reaction kinetics. Because now, instead of having just one pathway for methane formation, due to this incoming electrons and methanocyta is now able to activate two additional pathways for methane production. So there's three pathways actually working in tandem. So perhaps that explains why the overall kinetics is being enhanced through electron conductance by GAC, but also through the activation of the three methane formation pathways in methanocyta. So after we publish this paper, the next question is, of course, what is the mechanism or machinery responsible for taking up this electron. So this is something one of the PhD student that I'm co-supervising is actually working on, and we actually found something. So we're actually in the process of preparing a manuscript for this. So I'll just leave it there, but hopefully next time I can share more once the manuscript comes out. Okay, so how does then all this translates into actual applications or future impacts? So obviously this was a case where we were driven to use this approach because we saw some enhancement in the reactor performance. So we try to use this approach to explain what is happening at the microbial level. But of course, this can also help us generate some new potential applications and impacts in terms of redefining and enhancing the kinetics of sewage treatment, improving the calorific value of biogas. We can also think of, and I know there's also been work done by other research groups where they set up this kind of fuel cell system to directly do CO2 biomethanation using this kind of methanogens like methanol. So you can potentially think about energy offsets application using such kind of systems. And of course, the other area is to design new composite materials for maximizing electron harvesting, storage, and transfer. So that is the first topic. And now I would like to introduce to you a second topic, which is increasing bioplastic production. And the basis of this topic is really stemming from my PhD work. And during my PhD work, what I essentially did was I actually isolated a bacteria called Pseudomonas putida MBUS12. And this bacteria is quite special because it's able to utilize the plastic waste here and then turn it into bioplastic. And this bioplastic here is called medium chain length polyhydroxyalkanoate, or PHA. Of course, the conversion is not that direct. We actually had to work with another colleague that does a thermal process, and the thermal process basically melts this plastic waste into its individual monomers. For example, this is styrofoam. So the thermal process would melt this plastic into styrene, and then this styrene compound can then be utilized by Pseudomonas putida to make this medium chain length PHA. So this is a TEM photograph that I took of the bacteria, and you can see within the bacteria there are these white spherical granules. So these are actually the PHA granules. And then when extracted, it actually forms this kind of phloem. So why are we particularly interested in medium chain length PHA? Medium chain length PHA actually consists of carbon monomers here that are between 6 to 14 carbon. And this actually gives the PHA material a phloem-like structure, which is more pliable and stretchy. So it actually has some special applications. And the other thing about PHA is that it is both biodegradable as well as biocompatible. So it actually represents the best of both worlds in terms of the bioplastics that are out there. And of course, it has this advantage over conventional bioplastic in terms of its biodegradability. So the combination of biocompatibility and biodegradability means that PHA can actually be used for a variety of application, including a surgical application, pharma and biomedical application like drug encapsulation. It can also be used, of course, as food packaging or even plastic bags and so on and so forth. So the thing about medium chain length PHA is that if you look at the microscope at the population of genetically identical bacteria, you will see that not all of them have PHA, even though they are genetically identical. So you see some of them, they have the PHA here, but it seems to be completely empty in other bacterium. And in fact, medium chain length PHA production only occurs in 30% of the cell dry mass. And this is true for a lot of the organic substrates that's been tested for medium chain length PHA production in various bacteria, including Pseudomonas. So this is a problem. And this led us to ask a very simple question, but of course with very complicated implication. So our simple question is, why do genetically identical cells or organisms behave differently? So think about the idea of twins. This is a pair of mouse. They are twins, which means that they are genetically identical. But you can see that this mouse over here on the left is actually obese compared to this mouse that's on the right. So what is the factor at play here that is causing these differences between genetically identical organisms? And if we are able to know the answer to this question, then potentially we can have a strategy to manipulate the cells so that all the cells can behave the same way. And in the case of PHA production, it is actually to get all the cells to be producing this PHA so that we can push this PHA production to a higher number and helping the commercialization of medium chain length PHA. So this then brings us to this idea of quantum realities. I'm not sure how many of you watched this film by Michelle Yeoh, Everything Everywhere All at Once. And basically the premise of this film is that depending on the decisions that we make in life, our life can actually take different paths. So it's like, for example, in the film, Michelle Yeoh is in one path she is the lady boss of a laundromat. In another path, she is a movie star, and in the third path, she's a chef. And of course, the movie portrays a lot of other paths that she could end up taking based on her decision. So this is the idea that every decision we make can lead to a slightly different outcome, and eventually we can actually have many different outcomes based on critical decisions that we make at critical juncture. So this seems to mimic what we see in the cells, genetically identical cells behaving differently. So this led us to think that there is some similarities between the idea of quantum realities and gene regulation. So in fact, the physiological optimization of a biosystem is governed by gene regulation, whether a gene or a set of genes, whether they get turned on or they get turned off. And this pretty much would determine the phenotype that the cell would end up taking on. And there has been a lot of experimental data in complex biosystems, such as gene regulation, decision making, mutation, where they show that these processes actually violate classical information theory. So this leads us to the conclusion, or at least the idea, that gene regulation in a biosystem, a complex biosystem, does not follow classical rules, but in fact, it's more like a probabilistic quantum-like manner. So what I mean by probabilistic quantum-like manner is like, say, a guy meets girl and guy likes girl, so guy tries to propose to girl. And with this proposal, there is two possible outcome. One is the girl accepts the proposal, And the other equally likely outcome is that the girl rejects the guy. And based on this outcome, then it can take on different path. Um, Guy ends up marrying the girl, having a kid, not having a kid, and so on. Or guy tries a proposal again, succeeds, or he decides to give up and then he lives a lonely life. So these are all the possibilities that could come out from this singular event and the critical events that happen afterwards. So this is pretty much and each of this branch over here will come with a certain probability. So putting this into context of our PHA production over here, it is very widely known and also true in our case that PHA production in Pseudomonas putida is induced by nitrogen limitation. So this is a picture of the same bacteria um, under nitrogen-rich condition, and you can see that there is no white granules inside, so the cell doesn't accumulate any PHA. But once we lower the nitrogen levels, that's when the cell starts to accumulate PHA. So there's this two very distinct phenotype that we can see and we can induce through nitrogen availability. And if we start from a nitrogen-rich condition and we start to reduce the nitrogen level, then what we will see is that the PHA production will have this kind of curve-like phenomena. At conditions where it's nitrogen-rich, the cells doesn't produce PHA. And as we reduce the nitrogen level, it starts to produce more and more. And then there's a critical point where the nitrogen production will peak. And somehow after that, if we were to reduce the nitrogen levels further, the cell would go back to a state where they have no PHA production. So this kind of discrete peak is actually violating the total probability law, because the total probability law, classical total probability law, states that the bioplastic production should be a proportional function of the reduction in nitrogen. But this is, of course, not happening in our case. So we think that this is actually a bit more quantum-like and is probabilistic. And alongside with that, we have actually also collected information on the gene expression as well as the protein expression. And we actually found that during this process of shifting from non-PHA producing to PHA producing, we see that there is a differential expression of more than 4,000 genes, 2,000 proteins, and 100 over non-coding RNA. So there is actually a massive metabolic rewiring that is going on in this process. So therefore, our goal is really to kind of crack the code on this to understand what is actually bringing about this massive shift in metabolism. And if we can figure that out, then maybe we will have a way to get genetically identical cells to respond to nitrogen limitation in the same way, so that all of them will form PHA and we can increase PHA yield. What we did first is to consider this in a quantum probabilistic manner, where we use this generalized probabilistic theory to try to see whether we can use this approach to model this curve. So the mathematics behind it is a little bit complex, but I'll just try to share the conceptual idea behind this. So basically, we know that a cell, if it's in an initial state where it hasn't seen any nitrogen and it hasn't activated its pathways for PHA formation, then there is, so this is then what we call the initial state. And from this initial state, the cell can actually evolve to 4 different states, being the state where it detects a nitrogen limitation or it does not detect, and with each state, whether it forms PHA or it doesn't form PHA. So this is a 2 to the power of 2 kind of outcome. So this gives us 4 different outcomes. And with this information, we can then put it into this generalized probabilistic theory setup, where we say that you have a cell that is at state 0, initial state, and at state 1, it detects the nitrogen or it doesn't detect. And with this detection or non-detection, that could be an activation or non-activation of its metabolic rewiring. And then it gives us an outcome, that outcome being whether it forms PHA or not. And so this whole process of detection and activation can actually be coded in the form of quantum realm, allowing this state to transform from detection to activation and finally the measurement. So this is basically the same thing, the same representation as what I showed just now, where we have the cell initial state coded as a form of a matrix. And then we have the signal, which is signal D, which is the nitrogen level coded in another matrix. And then we allow this matrix to evolve into the detection state and further evolve into gene activation state. And then finally, we have the measurement, which is the probability of PHA production. So what we saw was that this method was actually able to predict the PHA production level quite well. So the blue dot over here is actually our experimental results. And then the orange line over here is the one that's predicted by the by this quantum-based approach. So this is actually very nice, and it just gives us this first indication that this kind of quantum-based approach can allow us to model this kind of behavior, system level, cell system level behavior. But of course, this doesn't actually answer the question of what is driving the evolution of the cell from the detection state towards the activation state. So this is the part that we are actually working on right now. And using traditional approach, like a lot of what our contemporaries, what they do is that we're able to actually map out what happens in the cell when we expose it to nitrogen limitation. So we were focusing very heavily on carbon metabolism, nitrogen metabolism, as well as PHA production. And what stood out to us was quite a stark picture. We actually saw that under PHA production state, the carbon metabolism was actually largely shut down. Okay, they are largely gray. Okay, and then the only thing that was going on was the PHA synthesis, which is this part over here. And another thing that stood out to us was the nitrogen metabolism. We actually saw a very active uptake of nitrogen, which was quite interesting to us because nitrogen was already in limiting amounts. So we then later found that the uptake of nitrogen was because the cell was using nitrogen and funneling that towards the TCA cycle for energy production. So the cell is actually switching the use of carbon to nitrogen for energy production. And all this seems to be sustaining the cell's production of PHA. But this production of PHA does not come from the carbon that is widely available in the system. It's actually coming from whatever that is already inside the cells. So this is quite insightful for us. And with this information, we then try to ask the question, okay, is there a way we can make the cell go in a way where we are getting this pathway still active all the way here? So if there's a way that we can do that, then we can get the cell to utilize all the external carbon and funnel them towards PHA production, and hopefully we can get more PHA. So Um, so this actually means that aside from just looking at what pathways are active and what pathways are not, we have to go and find what are the factors that are actually regulating the pathways. And it's not necessarily easy to see from this kind of metabolic pathway that is coming from only single layer of information, be it genomics or transcriptomics. In this case, we use 3 different layers. But the thing is that between these layers, there are actually additional proteins or genetic factors or even RNA factors that are regulating the information flow between these different layers. For example, DNA to mRNA, there are regulator factors that can activate the transcription or inhibit the transcription. From mRNA to proteins, this is the same as well. But all this information are actually missing from this kind of traditional approach that we are using. So the research challenge here is really that current approaches are very siloed and very manual. Siloed being that we are always examining them layer by layer, and it's a very manual process to piece together the information. And so we really need to start considering how these layers are connected and maybe look at them in terms of connection, in terms of layers and their connection rather than just singular layers. And the second challenge here is that the networks, if we were to consider the connections between these layers, then we can potentially end up with very large and complex networks that are computationally intractable in classical computers. So that is why we also started to look at whether there are quantum-based approaches that can help us tackle particularly the research challenge number 2. So fortunately, I managed to find a very brave PhD student who did the previous work. So now he's working on this. And we basically took the omics information that we have at the genomics layer, transcriptomics, and proteomics, and we started to construct the relationship between these different layers to form a graph or knowledge graph, if you will. So after we form the graph, we somehow have to find a way to figure out what part of the graph is actually important to answering our question, to figuring out the factors that are involved in PHA production. So what my PhD student did was that we used this method called a Gaussian boson sampling setup. It's a quantum setup. It's actually a quantum approach. And the simple way to understand this is that if you have a graph, so with the nodes and then the branches, the GPS approach is actually an approach where you shine light through the graph. So these are actually squiggly lines are actually light. And the part of the graph that is most densely connected, which typically means it's an important network, would end up transforming the light, either blocking it or squeezing it. And then this is how you end up getting some kind of output. So it will be able to tell you which part of the graph is actually dense and which part of the graph is not. And then we will be interested in the area that is dense. Okay, so there is a series of approach over here, but I'm not going to bore you with that. But basically, we are trying to look for a dense part of the graph. And then this is what we found. We found that in our bacteria, there are nodes which represents, each node actually represents a gene-protein pair, a gene and its corresponding protein pair. And we found clusters that are limited to only reach condition, limited to only the limiting condition, and existing in both networks. So there is definitely a very distinct metabolic state for the cell in the nitrogen-rich condition and the nitrogen-limiting condition. And among all this, we actually went through the nodes that were detected, and we largely see that these nodes adhere to known mechanisms about PHA production, particularly under nitrogen limited condition. We see that there is an increase in nitrogen transport and assimilation, but we also found other things we couldn't see from our manual curation. We found that there was ribosome hibernation and modulation, which means that the cell is not interested in making new proteins. And overall, we also saw a proteome maintenance. So the cell is somehow trying not to have too high a protein turnover, which I think is understandable because they don't have too much energy to spare for making new proteins. They are probably channeling towards conservation of their carbon sources and their energy in general. So we also found a connection to PHA production through the positioning of PHA granule proteins as important nodes across both conditions. So they are actually, these proteins are actually appearing in this area over here. And then as we were looking through, something popped out to us. We actually found uncharacterized a hypothetical protein that is very highly expressed as a protein. And it's actually at the center of the graph of this network, which is this protein. So this actually pipe of interest because it seemed to be right at the center, which means it's actually, and you can see it's heavily connected with a lot of the nodes over here, and of course here. And the circle is big, so it actually means that the protein expression, the relative protein and gene expression is actually high under nitrogen limiting condition. So we had to look into what this hypothetical protein was. And right now we could guess that it is this YCQI, YCG protein family. And there's not much known about it, but the predicted functional partners, and this is where it gets interesting, is that it is partnering with other proteins that are involved in amino acids export, assimilation, and also putative transcription regulators, which seems to fit this idea of nitrogen metabolism as well as transcription regulation control. So we think that this protein may have an integral role in PHA production and regulation. So actually what we're doing now is we are preparing to do the gene knockout to test the effect of this protein, this gene knockout on PHA production. So we'll see where that leads us. But my point is, using this kind of new approach, it can actually help us find things that we couldn't find before, that we wouldn't have known are actually important to this kind of metabolic shift processes. So we hope to further develop in this area. And of course, the goal is to be able to redefine and enhance bioplastic yield to help with MCL PHA commercialization, and also to develop a new quantum-based approach for data mining from complex omics data. And also some of our contemporaries and colleagues, they are working on quantum machine learning for environmental microbiome manipulation. And hopefully one day we will be able to really get the microbes to act in synergy to what we want, so that we can get them to produce certain desirable product on demand, and also increase bioenergy and bioresources production. That's my goal, but I think it will take us a while to get there. So that's the end of my presentation. Thank you so much for listening in, and I'm sorry if I overshot by quite a bit, and I look forward to our discussion.
Well, thank you so much, Amy, for this very exciting presentation. And now we have a few time for questions. So the online audience can also join the discussion. So it's open for everyone who would like to. I think someone already raised their hand. So who would like to start? Maybe questions?
Hello, thank you a lot for your talk. My name is Daniel. I'm also a PhD student from Valeria, and I was wondering that what you did to solve the kind of the problem to try to explain why the cells were behaving differently was through realities, and I was wondering if that is not a problem, not a problem, but that is not a possible explanation about stochastic processes, because you have like a deterministic process, which is the reduction of nitrogen, but then the answer of the cells kind of, I think they are kind of responding randomly. I don't know if it's actually random, but in this sense, my question is, do you have any idea that how you guys could measure this random factor?
Yeah, thank you for that question. And that's actually a very good question. So I'm actually working for this work that you saw. We are actually working with quantum information scientists. And what I learned from them through also our discussion is that there must be an explanation. You're right to say that this nitrogen limitation bringing about PHA production should be a deterministic process, and it should. The reason why it looks like it's stochastic is because there is too much that we don't know about the system. I mean, despite knowing a lot, there's still a lot that we don't know. And that is the problem. And this unknown here seems to be confounding our observations, or maybe sometimes even confounding what we know. And because of that, we have to find. So because of that, it kind of looks like it's stochastic. So the thing is, if we know everything about the system completely, then we should be able to model it using classical approach. If classical computers can do that, or we can find some good ways to reduce the computational resource, we should be able to do that. But we are at this point where there is a part that is unknown. And because of that, stochastic method is one way to take into account this unknown. Quantum approach is also another method to take into account this unknown. So that is why we are going into quantum approaches, because it has the added advantage of quantum computation advantage, particularly for complex network relationship. Yeah. But I think the long story short is, if one day we get to a point we know everything, then it shouldn't be a stochastic process. We predict that it shouldn't be a stochastic process anymore. I don't know if that makes sense.
No, it makes a lot of sense. I have never saw before the use of quantum the quantum theories to apply to microbial communities, and I think it is interesting. And I'm also thinking if what other areas of microbial communities we can apply this concept, because maybe this concept can try to answer questions that there are not, the answer is not just 0 or 1, is more quantum approach.
Yeah. So the well, through this process, what I found out is that the so the quantum information scientists, they have like a bunch of like models and algorithm, and it's absolutely critical because we are not quantum information scientists, so we don't have the ability to reinvent or invent new models and algorithm. So a lot of the use cases have to fit what they already have to apply those models and algorithm to do this calculation and analysis. And that's what exactly what we did. And to answer your question on what kind of scenario you can actually use quantum based approach, is like things like this, like where you have a can you guys see my screen?
Yeah.
Yes.
Okay. Yeah, things like this where you have this kind of discrete peak. You have a phenomenon like PHA production here, and it's associated with a discrete peak like this. Then this is one case where you can actually apply this kind of approach because it violates the classical total probability law. Yeah, and that's the general rule. Like any phenomenon that violates the classical total probability law, chances are you can apply a quantum-based approach. The other thing that we are actually showcasing here is anything that can be formulated as a graph, a knowledge graph describing relationships, describing interactions, can also benefit from using this kind of Gaussian boson sampling approach. In fact, this approach has actually also been applied towards image recognition, complex transportation network, and even protein 3D folding, which can be described as a graph. So this is another example. And I'm sure there is more, but these are the only two that I have more experience with. So that's as far as the extent I can share. Yeah.
Thank you a lot for the answers and highlighting this new approach.
Oh, thank you.
We have another question.
Hello, Amy. Can you hear me?
Yes.
Okay. I'm Christina. I'm a professor at the Federal University of São Paulo and also a postdoc researcher here in the group. And I'm going more or less in the same direction. Daniel was talking about the stochastic factors, and then I agree to a certain part that when we don't know much about a system, there is more to give to this stochastic factor. But then again, there's the emergent properties of a system. And that I don't know how much of it we could actually predict from knowing everything. But I think it's a very interesting approach. And do you think that this approach could help us to map, for example, not only what we don't know, but what properties could be an emergent property, and to try and find, because you're dealing with the production of PHA, and I'm thinking about also at the environment and thinking about the what happens and ecological interactions, or for example, even as methane as an emergent property. Do you think that this approach could also be used to map this kind of uncertainty?
That's a very interesting, but also certainly very important question, because I guess a lot of us in this field, we work with complex, or rather we work with mixed culture system, right? Because I think so far what I show for my approach, I've been using only pure culture. And I think the complexity will increase when we are dealing with mixed culture. And I think that's also where the possibility of having emergent properties will become even more prevalent in such system. I don't have a good answer for that, but I'm guessing we might have to involve machine learning, maybe quantum machine learning. So a hybrid use of machine learning as a black box to take into account that evolution of the system, the so-called emergent properties, into the main model itself, and the base model being very much still driven by knowledge and infused with the knowledge. So maybe it has to be of a hybrid, but I don't know how we're going to combine that. But I do know that quantum machine learning is something that people are already working on for environmental bioprocesses. And then the approaches I showed just now. So I'm sure there's a way to merge them, kind of like what we are doing now with the classical approaches, the machine learning and the empirical models. So if this can be done for classical, I'm quite sure it should be able to be done for quantum as well. But just this is my guess, long shot.
It's a very nice, thank you very much. And thank you very much for the great talk.
Hi, Amy. Thank you very much for your nice talk. Very inspiring and very mind-changing. I have a question about moving back to the first part of your talk regarding methanosylate, methane production. So you showed that it uses three simultaneous pathways for methane production, and they all are expressed at the similar levels. So do you have any clue why that happens? I mean, have you tried to knock out one of the pathway and see what happens then to the other two? Because it's interesting that it uses three pathways to produce methane, and are they thermodynamically different? So something like that.
Right. So we think that the reason why it of course we were a little bit disappointed to see that the acetoclastic pathway is still there. We're kind of hoping that it's less active, but the case is that it actually shows that it's equally active to these two other pathways. And we think the reason is because in the system, which is it's a sewage, it's a bioreactor treating sewage, right? So there is still VFAs that is going to be produced, and you still have acetate there. So I think that is the reason why this acetate pathway is still active. And for the CO2 pathway over here, we think that it is active because the electrons is being taken up by methanosylate, and upon its uptake, there are actually cell membrane proteins over here that can, and also cytoplasmic proteins over here that can transfer the the electrons to this electron carriers here, the NADP, the F420, and the ferredoxin. So we think that's why this system is able to utilize all three pathways at the same time. And we have not actually moved towards doing any genetic knockout of methanosylate. We do have a pure culture of methanosylate, but it's like notoriously difficult to grow. Takes like months to even get a culture going. So I don't think that would be an area that we will venture into. But certainly, I think this provides a lot of food for thought. And I think the genetic knockout can only further prove whether the methanosylate can, let's just say if you knock out the acetoclastic pathway, then I think it can further prove whether methanosylate can exclusively only use CO2 and electrons for methane formation. Yeah.
Okay, so basically it's using every resource available to produce methane.
Seems so. It seems to be that case, yeah.
Okay, thank you.
Based on your presentation, I already have some guests for my question, but I would like to hear from you. What would be the topics for the future that you would recommend for the young researchers like us and for the audience, which is the main part of our audience?
Learn quantum physics.
I think my answer is gonna be biased because of my background and my passion for microbes. But I think, just my personal opinion, I think if we are working with bioprocessors, then it is only logical and sensible that we try to understand the players that are taking part in this bioprocess. And if we can understand, I think that's when we can really combine with all the experience and the knowledge that we have from operating these processes to find ways to improve the operation, and maybe even make breakthrough to come up with new processes. I think that would be the first. So I think a combination of understanding how process work and the underlying biomechanism, I think, is important. The other thing is, and again, this is just my personal passion. I believe that if we are able to understand the tiniest unit of living organism, which is the bacteria or the archaea, it would also hold key towards understanding how complex organisms like ourselves function, because based on evolution biology, what they say is that we evolved from single living organism. And we do see a lot of the very important metabolic pathways like the TCA cycle are common across aerobic organisms, for example. So there must be some insights that we can get from this tiny microbes. And of course, I think the other part is also nowadays sequencing is very cheap and readily available, and we have a lot of this data and information. Are there better ways of utilizing this information? Bioinformatics analysis basically hasn't changed much in the past 20 years since I did my undergrad days. So that to me is something that I feel that there is a large area that's unexplored that we can potentially consider. And I think that could also come hand in hand to help us understand the mechanism, the biomechanisms of these microbes. So that's my take.
All right, thank you very much. Do you have any other question? I know that Professor Henry is very passionate about quantum research, and it seems that he contaminated you, and it seems that you contaminated many more here. So thank you very much for your presentation. It was really exciting, and we're very grateful. So,
Just check if there's no.
do we have any online questions? Let's just check here. Okay.
Okay.
So
So thank you, Amy. Thank you very much, and have a nice weekend.
you.
Starting now.
Thank you,
Amy.
everyone, for listening
Thank you for accepting
in.
our invitation.
Yeah, I wish everyone a good day ahead as well and a good weekend.
Yes. Have a good night. Thank you very much.
Bye-bye.
Thank you. Take care. Bye bye.